I'm going to be talking about ketogenesis and ketolysis. So, ketogenesis is formation of ketones. This only occurs in the liver. The liver is the only organ that can make ketones from scratch. And the way that this happens is by turning acetyl-CoA into a few different kinds of ketones. So we start with acetyl-CoA. Thiolase turns acetyl-CoA into aceto-acetyl-CoA. HMG-CoA synthase turns aceto-acetyl-CoA into HMG-CoA. HMG-CoA lyase turns HMG-CoA into acetoacetate. Acetoacetate turns into acetone, which is exhaled, or it turns into a more usable form of ketone known as beta-hydroxybutyrate using beta-hydroxybutyrate dehydrogenase. So we know dehydrogenase enzymes are typically paired with B3, and thiolase is paired with B5. That's the pathway of making ketones. So the three ketones that we make are acetoacetate, acetone, and beta-hydroxybutyrate. Like I said, beta-hydroxybutyrate is the most usable form. Obviously, we can't use acetone if it's being exhaled. So the liver makes these ketones, and it sends them, not really acetone again because it's exhaled, but it's sending these to other tissues where now these other tissues can break down those ketones and use them as energy through a process called ketolysis. So it's this same pathway but backwards. So we're starting here with beta-hydroxybutyrate. Beta-hydroxybutyrate turns beta-hydroxybutyrate dehydrogenase, the full name, using B3, turns beta-hydroxybutyrate into acetoacetate. HMG, these steps right here are not involved, so ignore these. Ignore this for ketolysis. So we're going from beta-hydroxybutyrate to acetoacetate, skipping over this, so acetoacetate to aceto-acetyl-CoA using thiotransferase. Then acetyl-CoA turns into acetyl-CoA using thiolase. So they're essentially almost identical pathways. It's just that ketolysis doesn't have this middle part with the HMG stuff. So the rate-limiting enzyme of ketogenesis is HMG-CoA synthase. The rate-limiting enzyme of ketolysis is thiotransferase. As I said in the beginning, the liver is the only organ that can make ketones, and it can't use them, though. It can't do ketolysis and use them as energy because the liver doesn't have thiotransferase, so it doesn't have this enzyme in the ketolysis pathway. So if it doesn't have that enzyme, it can't break down ketones. How do we enter ketogenesis? It can happen because of a few things, because of prolonged starvation, or when someone has severe diabetes mellitus type 1. Stimulators of this pathway, so acetyl-CoA will stimulate it because this is what we're putting into the pathway. If we have this, we can move forward. Glucagon stimulates ketogenesis because glucagon's the hormone that's high in fasting and starvation. And ketogenesis is happening in starvation. When fatty acids are being mobilized, that will also stimulate this pathway, but it's kind of upstream. So when we have fatty acids being mobilized, that means that there's being tags broken down in adipose tissue, and we'll have free fatty acids going to the liver and glycerol going to the liver. Glycerol is going to be used for gluconeogenesis, and fatty acids are going to be used for energy, and they're going to go through beta oxidation to turn into acetyl-CoA, and then that acetyl-CoA can be turned into ketones. This pathway is inhibited by insulin because insulin's going on in the fed state, and this is happening during starvation. G3P will inhibit this pathway. Basically anything in the synthesis pathway is going to inhibit this pathway because this pathway, again, is occurring in starvation. Organs that do ketolysis, which means they break down ketones into acetyl-CoA for energy, would be the brain during starvation, muscle, kidney, and heart also during starvation. And there's a clinical correlation here. So ketoacidosis is what occurs in prolonged starvation or severe untreated diabetes mellitus type 1, where there's no glucose reaching our cells, so we need to get energy from somewhere else. So we're doing gluconeogenesis during starvation. We're also doing beta oxidation, but acetyl-CoA is so high that we're going to start producing ketones from it. So like I said, this is the substrate of the whole pathway. So if we have this, we can go forward.